everyone, welcome to Agri Addict YouTube channel. I hope you all are doing well. So let's start with the lecture. So today I'll be discussing the third lecture of photosynthesis in plants in crop physiology lecture series. So this is my short intro. I have done my undergraduation from JNKV Jabalpur. Currently I am pursuing masters at IRI Hyderabad Hub Creda, Department Agronomy. I have secured All India rank 12th in ICR PG 2024 examination. So let's start with the lecture. In the previous lectures, we studied in the first lecture, we studied introduction part of photosynthesis. What is photosynthesis? What are the materials which are required in photosynthesis? Where photosynthesis is taking place? Everything we studied in the first lecture of photosynthesis. In the second lecture, we studied C3 cycle and photorespiration and all. Now we are starting with C4 cycle. It is also known as Hatch and Slack cycle because it is given by scientist Hatch and Slack. Okay, so let's start with the lecture. So first is, it is also known as C4 cycle. Why we are calling it C4 cycle is because the first stable compound which is formed in this cycle is a 4-carbon compound, which is oxaloacetic acid. Okay, so this is the first stable compound which is formed in this cycle. So that's why this cycle is known as C4 cycle. And this cycle occurs in C4 plants like maize, amaranthus, sorghum, sugarcane. What are the how these C4 plants are different from C3 plant? What is special in them? So they have dimorphic chloroplast, means they are having two types of chloroplast, okay, and they have kranz anatomy. This is very important. They have kranz anatomy and they have dimorphic chloroplast. Now comes the C4 plants have higher photosynthetic efficiency. How they have higher photosynthetic efficiency, we'll study. There is no photorespiration as we studied in the previous lecture, that is C2 cycle, photorespiration. This is an absolute waste kind of cycle. Here we are not producing any energy and we are not fixing any carbon dioxide. Even we are, whatever the energy we have already fixed, we are using that in this cycle, okay. And here in C4 plants, better H2O utilization is there. Okay, now comes this kranz anatomy. So what happens in C4 plants? Vascular bundle is surrounded by this specialized thick bundle sheath cells. Okay, this is the thick bundle sheath cells. So these are the importance or specialized structure. Why we are calling it as? Because the thing is they are thick walled and they are not having peptidase enzyme. They're having ribisco enzyme. Okay, this you have to remember. In bundle sheath cells, ribisco enzyme is found, but peptidase is not found. Means phosphoenol pyruvate carboxylase enzyme is not found in bundle sheath cells. In bundle sheath cells, they are having high number of chloroplasts, which are large in size and agranal, means they are not having grana in it. Okay, now comes the second kind of cell which is surrounded by which is surrounding the this bundle sheath cells is the mesophyll cells. These cells are thin walled, they are lacking ribisco. In this bundle sheath we studied that they are having ribisco enzyme but not peptidase. But in mesophyll cells, they are having peptidase enzyme but not ribisco. Here they are having less number of chloroplasts, and the chloroplast which is present is small size and is granal, means they are having grana in it. So what, as we studied, dimorphic chloroplast means two kinds of chloroplast. Morphologically, the chloroplast is different. How it is different? Here in bundle sheath cells, we are seeing, wait a minute. We are having larger size chloroplast and agranal chloroplast. Okay, this is a one kind. Here in case of mesophyll, we are having small size chloroplast which is granule, means they are carrying grana in it. Okay, so this is the basic difference between bundle sheath cells and mesophyll cells. So kranz anatomy is this arrangement of thick walled cell, that is bundle sheath cells, around the vascular bundle. Okay, now let's move to the next slide. Now comes the actual cycle. So this cycle was given by Hatch and Slack in 1967. Studied in detail in sugarcane and maize. They studied this cycle in sugarcane and maize, which is a C4 plant, leaves, and proposed a new pathway for dark reaction. The first stable product of this pathway is OAA, that is oxaloacetic acid, which is a 4 carbon compound. It is a 4 carbon compound, which is a dicarboxylic acid. Thus, Hatch and Slack pathway we call it, or C4 pathway, or dicarboxylic acid cycle. So first, let us see. This is the cycle. It is shown here. So this is the mesophyll cell. Okay, this mesophyll cell is attached with this bundle sheath cells. Now, what is happening? Whatever the atmospheric CO2 plant is fixing, it is getting inside the mesophyll cells. This mesophyll cell is combining with this phosphoenol pyravate PEP, which is a 3 carbon compound. Now this CO2 is fixing with the help of this PEP. It is getting converted into oxaloacetic acid, which is a 4 carbon compound. Now one NADPH, which is the product of this light reaction we studied in the second lecture. So this is the energy is used, which is getting converted into NADP. Now this by using this energy, this oxaloacetic acid is getting converted into malic acid and aspartic acid, which is also a C4 compound, means a 4 carbon compound it is. So this malic acid or aspartic acid, it is getting transported in bundle sheath cells. Now this malic acid and aspartic acid is going for decarboxylation, means what will happen? They'll release carbon dioxide. So here this NADP here we produced will be utilized and will produce NADPH2, and this will lead to the decarboxylation. This whatever the carbon dioxide we fixed here in mesophyll with the help of phosphoenol pyravate will get into the Calvin cycle. The main cycle which is operating here in bundle sheath cell is Calvin cycle. Okay, now after releasing this CO2, this malic acid and aspartic acid is converted into pyruvic acid, which is a 3 carbon compound. Now this pyruvic acid will be transported to mesophyll cell, and there will be a regeneration of phosphoenol pyruvate by using 2 ATPs. So here 2 ATPs are required for the regeneration of phosphoenol pyruvate for again fixation of this atmospheric CO2. This is the basic cycle. First, atmospheric CO2 is getting fixed with the help of this phosphoenol pyruvate. Here, peptidase enzyme is responsible for this fixation. Phosphoenol pyruvate carboxylase enzyme. It is responsible for fixation. The first compound which is being formed is oxaloacetic acid, which is a 4 carbon compound. Now one NADPH2 is used to convert that oxaloacetic acid into malic acid or aspartic acid. This malic acid or aspartic acid is getting transported to the bundle sheath cells. Now decarboxylation is taking place. So NADP is getting used and producing NADPH2. Release of CO2 is taking place. This CO2 will go to the Calvin cycle. Okay, and now the pyruvic acid formed, which is a 3 carbon compound, will get transported to the mesophyll cell for the regeneration of phosphoenol pyruvate, and this regeneration requires 2 ATP. So this is the basic C4 cycle. Okay, let's move to the next slide. So here the first CO2 acceptor in C4 plant is PEP, which is a phosphoenol pyruvate. It is a 3 carbon compound in mesophyll cell, while second CO2 acceptor is RUBP, which is a 5 carbon compound in bundle sheath cells, which is here in bundle sheath cells C3 cycle will be running. So Here you have to understood that in C4 plants also C3 cycle is running, but in C3 plants only C3 cycle is running. In C4 plants C4 cycles along with C3 cycle is running. Okay, so in bundle sheath cells C3 cycle is running. So here 5 carbon compound RuBP is the second carbon dioxide acceptor. First carbon dioxide acceptor in C4 plant is phosphoenol pyruvate. Initial fixation of CO2 in mesophyll cell is catalyzed by this peptidase enzyme, phosphoenol pyruvate carboxylase enzyme, which results in the formation of oxaloacetic acid, which is a 4-carbon compound. Then, reduction of oxaloacetic acid takes place by NADPH2, results in the formation of malic acid, or transamination of oxaloacetic acid result in the formation of aspartic acid. Either malic acid or aspartic acid will be formed by oxaloacetic acid. The malic acid or aspartic acid formed in mesophyll cells is transferred to bundle sheath cells. In bundle sheath cells, the oxidative decarboxylation. This decarboxylation means removal of carbon dioxide. So from malate, which is a 4-carbon compound, this decarboxylation will take place. CO2 will be released and this will result in the formation of pyruvic acid. And this will go for regeneration of phosphoenol pyruvate, which is a 3-carbon compound. Okay. This released CO2 in bundle sheath cells is accepted by RuBP catalyzed by Rubisco, which is a C3 cycle which will be taking place. The C3 cycle or Calvin cycle operates in bundle sheath cells with utilization of 18 ATP and 12 NADPH2, which we studied in the previous lecture also. That to run a whole one Calvin cycle for production of one glucose, we require 18 ATP and 12 NADPH2, resulting in the formation of glucose. Okay, pyruvic acid from bundle sheath cell return to mesophyll cells. It regenerates the PEP, which is the primary carbon dioxide acceptor, by utilization of 12 ATP, catalyzed by enzyme pyruvate phosphate dikinase. Okay, so in C4 plants, total 30 ATP is required. 12 NADPH2 is required for one glucose formation. See, in this one cycle, one carbon dioxide is getting fixed. Okay. Here, 2 ATP is required for that regeneration. And here, one C3 cycle is also running, right? This C3 cycle we earlier studied, it requires 18 ATP plus 12 NADPH2 for fixing one glucose. Okay. Now this one carbon fixation require 2 ATP. So if we want 6 carbon dioxide fixation, it will require 6 into 2. Means 12 ATP. Right? Now you add this 18 ATP which is required in C3 cycle plus 12 ATP. How it will be? It will be 30 ATP. So this is how we got this 30 ATP. So C3 plants, we require 30 ATP, and whatever this NADPH2 is being required in C3, it is the same, because in C4 cycle, whatever the NADPH2, one molecule of NADPH2 we are using, and we are again producing it in bundle sheath cells. So it is getting, so there is no net use of NADPH2 in C4 cycle. Whatever the NADPH2 we are using in C3 cycle, that is the only NADPH2 required in C4 plants. Okay, so total 30 ATPs and 12 NADPH2 are required for the synthesis of one glucose in C4 plants. Okay, let's move. So, as for the C3 cycle also we studied. Now for C4 cycle, for fixation of one carbon dioxide, 5 ATP is required. 3 in case of C3 cycle and 2 in case of C4 cycle, and 2 NADPH2 is required. So for fixation of 6 carbon dioxide, 6 into 5 would be 30 ATP, and 2 into 6 will be this 12 NADPH2. Okay. Let's move to the next slide. Now comes the CAM pathway or cruciation acid metabolism or dark carbon dioxide fixation. This CAM pathway was discovered by Oliari and Rouhani. This is a scientist name and you have to remember. In the C4 plant, the C4 cycle was discovered by this Hatch and Slack, and this CAM pathway is given by Oliari and Rouhani. So scientist name you have to remember. They observed that carbon dioxide fixation occurs during night in the members of Chrysolaceae family or succulent xerophytes, desert plants. Okay, succulents or CAM plants are characterized by photoactive stomata. They have different kind of stomata. So photoactive, this word you have to remember. What does it mean? Is these plants, they open their stomata during night and remain closed during daytime. They open their stomata during night time to minimize the transpiration loss. Okay, what they do is they fix the carbon dioxide in the form of acid in night, because the stomata is open during night only. So during that time they will fix the carbon dioxide in the form of acid, organic acid, and they will utilize it in the morning time when the light will be there. So because in the daytime the stomata is closed. So let's study this C4 cycle. Now this carbon dioxide, as the stomata is open in case of, this is night time. Okay, in night time they are opening the stomata, carbon dioxide is getting inside. This carbon dioxide is getting fixed using this peptidase enzyme and PEP compound, as we studied in C4 plants also. So this is they are converting it into oxaloacetic acid, a 4-carbon compound. This oxaloacetic acid is converted into malate or malic acid by using one NADPH2 molecule. Okay, this malic acid is getting stored in the vacuole of plant during night time. So this is what is happening in night. Stomata is getting open, CO2 is coming, it is getting fixed with the help of phosphoenol pyruvate, in presence of phosphoenol pyruvate carboxylase enzyme. Now it is forming oxaloacetic acid, 4-carbon compound, with the utilization of one NADPH2. This is NADPH2. There is some error here. This should be NADPH2. Okay, one NADPH2 it is using, converted into malate or malic acid, and this malic acid will be stored in vacuole. Now comes the daytime. And in daytime, the stomata is closed. No fixation of carbon dioxide will take place. So whatever the malic acid which is stored in night in vacuole will come outside. This malate will utilize one NADPH, where will get converted into NADPH2, and this will release one carbon dioxide. Here carbon dioxide removal is taking place. So this is decarboxylation. This is the decarboxylation process. And now this malate is getting converted into pyruvate or pyruvic acid, which is using one ATP here, and getting this PEP is formed. Regeneration. This method is regeneration. Here this regeneration of PEP is taking place. So first, in night, stomata is open, carbon dioxide is fixed using this PEP, 3-carbon compound, in presence of PEP carboxylase, getting converted into oxaloacetic acid. This oxaloacetic acid is getting converted into malate or malic acid using one NADPH2 molecule, and this malic acid is stored in vacuole. This is the night work. Okay. Now comes the morning shift. In morning, the stomata is closed. Now whatever the malic acid which is stored in vacuole will come out from vacuole in cytoplasma, and now this malate will get converted into pyruvate by releasing one carbon dioxide, and this carbon dioxide will go in the chloroplast and will get involved in the Calvin cycle. So Calvin cycle is a kind of universal cycle. It is taking place in C3 plants also, C4 plants also, and CAM plants also. So here the main work which is being done was by this Calvin cycle only. whatever the CAM plants is doing is they are fixing the CO2 in night, because stomata is open during night only. So this is the concept for CAM plants. Let's move to the next slide. Now I have simplified this using this. So this is the scenario. This is the mesophyll cell in which it is taking place. So this is the night scenario and this is the day scenario. In night, what is happening? This carbon dioxide is coming from outside, from atmosphere, is getting this fixed by this PEP, 3-carbon compound, in presence of this PEP carboxylase. So this is forming this oxaloacetic acid, which is a 4-carbon compound. This 4-carbon compound will get converted into malic acid or aspartic acid, and this acid will get stored in the vacuole in night, during night. Okay. Now, during daytime, when the stomata is closed, this malic acid will come out. This will release this CO2, and this CO2 will go for C3 cycle, and now it will result in the formation of pyruvic acid, which is a 3-carbon compound. This pyruvic acid will go for the regeneration in presence of PEP synthase enzyme, and this enzyme will require 2 ATP plus half ATP. So it will require 3.5 ATP in the regeneration. In the previous C4 plants, we studied that this PEP synthase is requiring only 2 ATP, but here it is requiring 3.5 ATP. So more ATP is being consumed for the regeneration. This is the regeneration process. Regeneration of PEP. Okay, so this is the whole scenario. Here, because C3 cycle is running, so 3 ATP and 2 NADPH2 as it is required in C3 cycle. This is the requirement. This is constant. So, rubisco and pepcase are present in mesophyll cell. In this case, rubisco and pepcase both are present in mesophyll cells. Okay. Now comes the CAM cycle. So, for CAM cycle, for fixation of 1 carbon dioxide, 6.5 ATP is required. How? This in C3 cycle we are requiring 3, and this for regeneration we are requiring 3.5. So 3.5 plus 3, it is 6.5. So 6.5 ATPs are required for fixation of 1 carbon dioxide and 2 NADPH2 as in case of this C3 cycle. So 6.5 ATP plus 2 NADPH2 is required for fixation of 1 carbon dioxide. Now comes for synthesis of 1 glucose, we require 6 carbon dioxide. So the cycle has to run 6 times. So 6 into 6.5, that is 39 ATPs are required for 1 glucose synthesis and 12 NADPH2. This you have to remember. For Calvin cycle, it is different. It is 18 ATP. For C4 cycle, it is 30 ATP, and for this CAM pathway, it is 39 ATP. Okay, so that NADPH2 number is constant for all the 3 cases. Now comes CAM plants like Kalanchoe, like Bryophyllum, Opuntia, Agave, Aloe, Euphorbia, Pineapple, Welwitschia, or this is a gymnosperm. Okay, these plants are adapted to water conservation. The primary acceptor of carbon dioxide in this case is PEP, and oxaloacetic acid is the first compound. Pepcase and rubisco both are present in the mesophyll cell. But in case of C4 plant, it is different. Okay. Next, let's see a summary. So, for fixation of 1 carbon dioxide in C3 cycle, we need 3 ATP and 2 NADPH2. Whereas for C4 cycle, we need 5 ATP and 2 NADPH2. In case of CAM pathway, we require 6.5 ATP and 2 NADPH2. So for fixation of 6 carbon dioxide or synthesis of 1 glucose, we need 18 ATP, 12 NADPH2 in case of C3 cycle, 30 ATP and 12 NADPH2 in case of C4 cycle, and 39 ATP and 12 NADPH2 in CAM pathway or CAM plants. Next, now comes the differences between C3 plants and C4 plants. Cell type in which Calvin cycle takes place. In C3 plants, Mesophil. In mesophil only the C3 cycle is taking place or Calvin cycle is taking place. But in C4 plant, this Calvin cycle is taking place in bundle sheath cell. This is important. The cell type in which the initial carboxylation reaction occur. Here in C3 plants also initial carboxylation reaction is taking place in mesophil. In C4 also it is taking place in mesophil. How many cell types does the leaf have that fix CO2? In case of C3 plants, we are having only one cell, that is mesophil. But in case of C4 plant, we are having two, that is one is mesophil and second is bundle sheath. Okay. Now, which is the primary carbon dioxide acceptor? For C3 plants, primary carbon dioxide acceptor is RUBP, where in case of C4 plant, it is PEP. Okay. Number of carbons in primary carbon dioxide compound acceptor is 5. This RUBP is a 5 carbon compound, whereas this PEP is a 3 carbon compound. Okay. Which is the primary carbon dioxide fixation product? So in C3, phosphoglyceric acid, which is a 3 carbon compound, this is the first fix, like that's why we are knowing C3 cycle as C3, because the first stable compound which is formed is a 3 carbon compound, whereas in case of C4 plant it is a 4 carbon compound and this is oxaloacetic acid. Number of carbons in the primary carbon dioxide fixation product. So it is a 3 carbon compound and OAA is a 4 carbon compound. Let's move to the next slide. Does the plant have rubisco? Yes, of course. In C3 it is having rubisco. In C4 also it is having rubisco. But this rubisco is present in bundle sheath cells. Okay. Does the plant have pepcase? In case of C3, yes, it is there, and in case of C4 also it is there. Which cells in the plants have rubisco? Here in C3 plants, mesophil is having rubisco, and here in C4 plants, as I said earlier, bundle sheath is having rubisco enzyme. Carbon dioxide fixation rate under high light condition, here it will be medium in case of C3 plant, but for C4 plants, it will be high. Okay, these are more efficient in fixing carbon dioxide under high light condition. This you have to remember. Whether photorespiration is present in low light intensity? No, because the condition for photorespiration is high light intensity, and high temperature. In C4 also during this low light intensity, no this photorespiration. In case of C4, there is no photorespiration, by the way. There is no photorespiration mechanism. Okay. Whether photorespiration is present in high light intensity in C3 plants? Yes, it is there. In C3 plants, if the light intensity is high, high temperature is there, C2 cycle or the photorespiration will take place. Whereas in C4 plant, there is no such mechanism. Whether photorespiration would be present in low carbon dioxide concentration? Yes, in case of C3 plant it will be there, but this photorespiration is not there in case of C3 plant. C4 plants. That's why we are saying the photosynthetic efficiency of C4 plants is high. Okay. Whether photorespiration would be present at high carbon dioxide concentration? No. If the concentration of carbon dioxide is more, so rubisco's carboxylase part will be working with very high efficiency. So there will be no photorespiration. And C4 plant, there is no photorespiration, as is I said earlier. So temperature optimum for this C3 plant is 20 to 25 degrees Celsius. Whereas in case of C4 plant, because they can survive at higher temperature, so it is 30 to 40 degrees Celsius. What are the examples of C3 plants? This is wheat and rice, and here maize, sugarcane, sorghum. Oh, okay. I hope you have got the difference between C3 and C4 plants. Okay. Now comes the factors affecting photosynthesis. So there can be two kinds of factor, internal factor and external factor. So what are the internal factors? So first is assimilatory number. What is assimilatory number? It is how much carbon dioxide fixation is done by 1 gram of chlorophyll in 1 hour. This you have to remember. Assimilatory number. How much of carbon dioxide fixation is done by 1 gram of chlorophyll in 1 hour. Okay, so it depends on assimilatory number. If the assimilatory number is high, the photosynthesis rate will be high. Okay, now comes the genetics. The amount of chlorophyll will be present. If a plant is having low amount of chlorophyll, then the photosynthetic rate will be slow. Okay, if the number of chloroplast is high, photosynthetic rate will be high. The number of leaves is high, so photosynthesis rate will be high. Now comes the external factor. So let's discuss the external factor. So first factor, and that's external factor, is the light. So there is a linear relationship between incident light and carbon dioxide fixation rate at a low light intensity. When the light is low, here the light is low, so we are saying that there is a linear relationship. Means if we are increasing the light intensity, the rate of photosynthesis is increasing, but only up to a certain level. Means here this low light condition, medium light condition, and now comes the high light condition. Okay, so in low light condition, we are seeing that the rate of, when we are increasing light intensity, the rate of photosynthesis is also increasing with the linear way. Okay, at higher light intensity, gradually the rate does not show further increase as other factors become limiting. Light saturation occurs at 10% of full sunlight. So at this point of time, this saturation is taking place. Beyond that, if we are increasing the light intensity, the rate of photosynthesis is not increasing, it is becoming constant. Okay, so except for plants in the shade or in dense forest, light is rarely a limiting factor. So generally light is not a limiting factor for plants, but yeah, it is an external factor which can influence plant. Okay, increase in incident line beyond a point causes the breakdown of chlorophyll. If we are increasing light intensity, the photosynthesis will not be increased. Okay, this will be constant, but if we are keep on increasing the light intensity, this will lead to the breakdown of chlorophyll. If the chlorophyll will be breakdown, there will be a decrease in photosynthesis rate. Okay, this is important. Next comes the temperature. The dark reaction being enzymatic at temperature control. Okay, dark reaction, because we are using this peptidase enzyme, ribisco enzyme. These are the enzymes which are being used. So enzymes are temperature controlled. Okay, so if the temperature is increased very much high, so this will become denatured, because all enzymes are protein. The protein will get denatured and the functioning will not be there. So though the light reactions are also temperature sensitive, they are affected to a much lesser extent. Now, the C4 plants respond to higher temperature, that is 30 to 40 degrees Celsius, show higher rate of photosynthesis. Even if the temperature is near about 30 to 40 degrees Celsius, that is a high temperature, C4 plants will show higher rate of photosynthesis there, because they are adapted in that condition. Whereas in case of C3 plants, a much lower temperature is optimum. Means they can function properly within this range, that is 20 to 25 degrees Celsius. Now, tropical plants require higher temperature optimum, that the plants adapted to temperate climates. So tropical plants are adapted to high temperature, so they require high temperature optimum. Okay, let's move to the next slide. Now comes the carbon dioxide. So carbon dioxide is a major limiting factor for photosynthesis. This is important. It is a major limiting factor. The concentration of CO2 is very low, and in atmosphere it is between 0.03 to 0.04 percentage. Currently it is 419 ppm. Okay, this is 419 ppm. This is the parts per billion concentration. So the C3 and C4 plants respond differently to carbon dioxide concentration. At low light condition, neither group respond to high CO2. If the light intensity will be low, neither C4 nor C3 will respond to the high carbon dioxide condition. But as the at high light intensity, both C3 and C4 plants show increase in rates of photosynthesis. At high light intensity, both C3 and C4 plant will show response, and there will be an increase in rate of photosynthesis. In case of C4 plants, they show a saturation at about 360 ppm. At lower concentration of carbon dioxide, the C4 plants get saturated. Okay, so they can achieve maximum rate of photosynthesis at lower concentration of carbon dioxide. Whereas in case of C3, they respond to increased carbon dioxide concentration, and saturation is seen only beyond 450 ppm. So they require high concentration of CO2, that is 450 ppm, to get a saturation level or a maximum photosynthetic rate. So we can say that current availability of CO2 levels is limiting to the C3 plants, but not for C4 plant, because C4 plants, they are getting saturation at 360 ppm, but for C3 plants, the saturation point will come only after this 450 ppm. So so carbon dioxide is limiting in case of this C3 plants, current carbon dioxide concentration. Now, atmospheric carbon dioxide is not limiting factor for C4 plants and submerged plant. It is limiting for C3 plants. Now comes the carbon dioxide compensation point. What is carbon dioxide compensation point? It is the point where rate of photosynthesis become equals to the rate of respiration. What is happening here? The rate of photosynthesis is equal to rate of respiration. If one glucose is synthesized, this one glucose is getting breakdown, is producing ATP, and this ATP is utilized by plant. This is the scenario at carbon dioxide compensation point. Rate of photosynthesis is equal to rate of respiration. So there will be zero net primary productivity. This is important. For C4 plant, the carbon dioxide compensation point is 0 to 10 ppm. Whereas for C3 plant, it is 25 to 100 ppm. This value you have to remember. Carbon dioxide compensation point for C4 is 0 to 10 ppm, whereas for C3 plants, it is 25 to 100 ppm. Let's move to the next slide. Now comes the water. Even though water is one of the reactant in the light reaction, the effect of water as a factor is more through its effect on plants. See, if water will be less in plant, the plant will show wilting rather than directly on photosynthesis. So photosynthesis will be affected later on. First, the plant will show wilting, the movement of nutrients and water will not be there. Okay, so the stomata will get closed. So it is having indirect effect on photosynthesis. The water stress causes the stomata to close, which will reduce the carbon dioxide availability. If the carbon dioxide availability will not be there, ultimately the photosynthesis rate will decrease. Besides this, water stress also makes leaves wilt, thus reducing the surface area of the leaves and their metabolic activity as well. Okay. Now comes the questions. So first question is, for every carbon dioxide molecule entering in the Calvin cycle, required energy is. Calvin cycle is nothing but C3 cycle. And in C3 cycle, in the previous class we studied, we need 3 ATP and 2 NADPH2. So correct answer is first 3 ATP and 2 NADPH2. Okay, let's move to the next slide. Most abundant enzyme in the world is. So most abundant enzyme in the world is, you have to remember, is ribisco. Okay, this is important. Now comes the phosphoenol pyruvate is the primary carbon dioxide acceptor in. So C4 plants, C2 plants, C3 or C4 or C3. So PEP is primary carbon dioxide acceptor in C4 plants. Okay, thank you for watching the lecture. I hope you have understood this photosynthesis in plants concept. So if you have any doubt in this concept, you can comment, and I'll definitely try to solve your doubts. And all the best for your examination, and keep studying. Thank you.